different areas of domains. So we thought that would be a very interesting topic. And then we put up a proposal to AICTE and ISTE essentially. And then they accepted that essentially. And that was almost a year back before the pandemic started. And now we are in this very different time zones essentially, where we are into this pandemic. It's a once in a lifetime event that we all are seeing essentially. So now everybody is talking about drug development. And so interestingly, even the watchman of my building is also talking about drug development. He on and off, he talks to me about, what are the medicines being made, and even he has an opinion on that. Right. So drug development essentially, as we all understand as educators, is fairly wide in terms of the domain. It's very multidisciplinary in the nature. So, and that is how we've tried to put together this 1 week faculty development program. So obviously, you cannot cover the entire aspects of drug development in a 1 week program. But we've tried to touch the domains of it essentially. So right from starting from more of a preclinical approach essentially to more of a clinical approach, and then fusing it all together essentially. So that was our objective behind that, and we've spoke to our speakers, and we've really got them on board with us. So today, as you see here essentially, so we have 2 speakers essentially, and we have Dr. Farooq essentially, who's been talking about some very exciting work that he's been doing at Monash, primarily with regards to using a Zebrafish model as a very innovative disease model essentially. So very exciting work, a very young researcher, dynamic researcher, very interesting thought. And post that, Dr. Abdul Basit comes in from University of Washington, and he'll be talking more about drug transporters essentially. And transporters are a very newer and emerging and exciting area where a lot of variability that happens into humans essentially. When you look at the drug exposure, when you talk about PD differences, pharmacodynamic differences, a lot of that can be attributed to transporters. So there's a lot of work going on transporters. So he'll be sharing some of this work that he's been doing at University of Washington. So that, we're moving from more of a preclinical space. We're talking about innovative disease models. And then tomorrow, then Dr. Dafiz comes in from Monash University. He'll be talking more about optimizing the drug targets, or using more of a molecular docking methodology to optimize the drug targets, more of a rational based drug design as such. So, getting the right targets, hitting the optimal targets is key to start with a good drug essentially. Beyond that, on day 3, we start to go more of a preclinical clinical space essentially. We'll have Dr. Mansi coming in from US, and she'll be talking more about biomarkers. So biomarkers has been a very exciting field. There's a lot of work going on to biomarkers in different areas of drug development, right from patient selection to optimization. I remember some years back in 2012, when we were putting up a proposal for one of our oncology asset, PD-1, to FDA. FDA was very interested in knowing which are those patients that are likely to get benefit from this drug. Is there a biomarker that can really differentiate? Because what the data that we were seeing was very dichotomous, in the sense that we were seeing some patients who were dramatically responding back to the drug, and there were some patients who did not respond to the drug. So FDA was very keen in knowing that whether do we have any biomarkers that would help us identify those patients who were, number one, able to respond, and second, because it was an oncology drug, you have to follow the patients all the way to the recovery or unfortunate event of death. So if it's a death as an event, you really have to follow them very long, essentially. So is there a good prognostic marker that you could tell us, that would tell us that these patients are not going to run? So, stuff like that. So Dr. Manasi will come in day after tomorrow on day 3. She'll be talking about some of the exciting work she's been doing with biomarkers. And then day 4, another exciting area of, which is something which is close to my heart, is basically use of modeling and simulation for drug development as such. So we will have a whole panel of experts, right from our very own Dr. Iyer, who is a professor at Bombay College of Pharmacy. He'll be talking about what models are, what our PK and PD basically models are, essentially. And from then there on, we have 3 speakers who are coming from BMS USA, who are using these methodology of modeling and simulation into optimization of drug development. So Dr. Kinjal comes in early on. Well, she'll be talking about how our models used up in early phase of drug development, right from dose selection, right from patient population, some of the very exciting work that they've been doing at BMS. And then Dr. Amit Roy comes in, and he talks about some of the work that they are doing in the late stage development. So Amit is a global thought leader into pharmacokinetics and pharmacometrics, and he's led several drug applications in US, and he was the one who was a part of the new drug application that was for Epilimumab. So Epilimumab was the first drug that was approved into immuno-oncology asset, and that was the drug that Dr. James Allison, who got the Nobel Prize a year back, he was the one who had pioneered that area of immuno-oncology. So Amit will talk a lot about how his modeling and simulation helping him in the late phase designs essentially. And Dr. Mike Sense comes in at the late essentially. He's a very senior industrial expert. He also works at BMS. So he'll be talking in general about his overall experience of modeling simulation. Incidentally, Mike Sense was for 10 years at the Bangalore facility of BBRC essentially. So BMS has a collaborative facility with Biocon. So Biocon BMS Research Center, they do a lot of early phase drug development. So that was on day 4th. And after that, on day 5th, Dr. Tahir comes in. He talks a lot about new drug delivery methodologies. And on the final day, we have some very exciting speakers who would tie everything down together essentially. So we have Dr. Rana Rais, who comes from Johns Hopkins University. She's an associate professor in neurology as such. Also happens to be my junior at University of Maryland. So she's heading the drug discovery lab there. So she'll be talking about some of the assets that they have carried all the way from preclinical to clinical and ultimately out licensing that into the market. So end-to-end experience of the drug development. Along with her is Dr. Kafil, who's the director and the CEO of Resino Biotech. Is a very experienced person to biotechnology and is doing some very cutting edge science with developing antibodies towards COVID. So we've tried to cover the entire spectrum of drug development so that it gives you a good flavor about how the whole entire process is. And we all understand, drug development is not an easy process. It's a fairly lengthy process. It's a grueling process. It is very brutal in the sense that the number of asset that comes into preclinical from there all the way to what gets approved, there's a lot of attrition that happens, and thousands and hundreds of thousands come into the start, and only one ends up as a drug as such. So it can be brutal. It's time consuming. It is expensive. A couple of years back, we were working on an oncology asset for liver cancer. We took it all the way to phase 3 trials, and at that point, we found out that drug is not working. So imagine the amount of time and money that went into the almost $1 billion plus of shareholder value went out with no returns as such. So it can be very frustrating as such. So the science has to be very innovative. FDA has a very critical path initiative where they talk about how can we make this drug development process very innovative in terms of the target selection, in terms of the clinical trials, in terms of how optimally we can do that. So that we can get the best of the therapies to the people in a cost-effective manner, in a time-effective manner to all essentially. So that was the whole idea behind that. So we have tried our best essentially to give a good comprehensive look to this entire FDP, doing justice to the title that we have essentially. And I really appreciate the whole lot of speakers who have really agreed. All of them have been extremely busy, but they have really agreed to come to this FDP and talk about their research. For some of them who are speaking from the other end of the globe, it is very early in the morning. It's like 5 and 6 a.m. in the morning for US folks. So, but they're excited to come in and talk to you all essentially. So that's what it is essentially. I know we all are suffering from a syndrome which is called as webinar syndrome. So everybody, with this whole pandemic, too many webinars. There's something called as webinar fatigue that has set into. But I can assure you that we have very carefully designed the entire program, and hopefully the hour and 2 hours, whatever you spend on everyday basis in the evening for the entire week, would be well spent essentially. So with that, I really welcome you once again to a campus. And some of you who don't belong to Mumbai and they're logging from outside, if at all you come to Mumbai, do visit our campus. And on behalf of the entire management and the School of Pharmacy at Anjuman-I-Islam's Kalsika Technical Campus, I once again welcome you to this AICTE-ISTE workshop for the entire week, essentially. I hope and I wish you a very enjoyable learning week. With that, welcoming once again. Over to you, Professor Basukyan.
Thank you, Dr. Sharik, for welcoming our gathering. Going ahead, we have with us Dr. Abdul Razak Hanutagi, who is a founding director of Anjuman-I-Islam Kalseka Technical Campus. He did PhD from IIT Bombay. He was former principal of MS Sabu Siddiq Polytechnic, Mumbai. During his tenure, Sabu Siddiq Polytechnic was adjudged as the best polytechnic of the Maharashtra state in the year 2000 and role model polytechnic during 6th ISTE annual convention. He has more than 32 years of professional experience in the field of engineering education and research. He is a recipient of Outstanding Research Paper Award in the International Engineering and Technology Educational Conference held at Taylor University, Malaysia. He is a recipient of Silver Jubilee Memento for his dedicated service at Basveshwar Engineering College. He is a recipient of Best Teacher Award, Anjuman-I-Islam, and citation from Lion Club International, Mumbai for his distinguished services to the society. He is also recipient of Lokmat Panvel Gaurav Puraskar, organized by Sri Ram Sheikh Thakur Social Organization Panvel. As a part of this technical campus, I proudly say that Dr. Abdul Razak is a humble leader who developed a vibrant team, and Kalseka Technical Campus was conceived by him. With this brief introduction, now may I request our director, Dr. Abdul Razak, to introduce our parent organization to all the external delegates. Sir, it is over to you.
Thank you, Professor Abu Sufyan, and also thanks to Dr. Sharik Syed, who put the entire, context of this STTP. I once again join in welcoming all delegates, all participants for this well-thought short-term training program, exclusively designed for the benefit of faculty members of pharmacy. Welcome, and I will not take much of your time because I am odd man out, as related to this program. Yes, as a director, my responsibility is to at least briefly introduce our organization so that the external candidates may get a little bit about our organization. So that is my responsibility. So thanks for this opportunity, Professor Abu Sufyan. Can I get screen sharing?
Yes, sir, it is permitted. You can go ahead.
Okay, fine. I think, I don't know, how many of you know about Anjuman-I-Islam, especially external candidates who have joined this program from various parts of the country. We are really very proud to be associated with this organization named Anjuman-I-Islam. There is some misconception assuming that it is something related to some Islamic activities by name. Actually, Anjuman-I-Islam is a educational and social organization set up well before our freedom. So if you can trace, it got established in the year 1874. Of that time, by leading illuminaries like late Justice Badruddin Tayyibzi. He was a highly educated personality of that time, and he was, in fact, first Indian judge of Bombay High Court. That kind of person, along with two of his other colleagues and his brother, they thought of a noble idea of educating Muslims in particular, and all the communities, all people who are relatively backward in education. So with that notion, they started Anjuman-I-Islam, a small school with 3 teachers, 120 students, way back in 1874. Then since then, it has grown today almost like an ocean. And for your, in at least remembrance, I would like to highlight certain key points. As I said, Anjuman-I-Islam was the main center for freedom struggle activities. Most of our founders, they even went to jail at the time of freedom fighting. So that way, Anjuman-I-Islam is basically established by freedom fighters of our country. And accordingly, they set objectives for educating, all backward classes and Muslims in particular, minorities. And by now, Anjuman-I-Islam is standing tall with 97 institutions and around 110,000 students under its portals. About in the year 2000, even many a times, even it was awarded as best educational organization by government of Maharashtra. And we run various courses. We say very popularly, our journey in Anjuman-I-Islam starts from kindergarten to KG to PG to PhD. So you name, everything is there except the traditional MBBS program. But we have got BUMS, Unani Medical College. So you name any of the educational program, this Anjuman-I-Islam has under its ambit. So today it is standing as the largest educational organization for minorities in the country. And even it is larger than Aligarh Muslim University, which got established a year after Anjuman-I-Islam. It was established in 1875. So we are really lucky to be associated with this organization, wherein a true service to the field of education is happening. And not only for the sake of educating people, but it is also taking care of marginally challenged section of the society. And though it is a minority institution, but its doors are open for all. It is truly holding secular credentials. It is practicing the secular values since its establishment. And therefore, you find the entire mini India under the portals of Anjuman-I-Islam. Be it students, faculty, and staff, you will have everybody. So under Anjuman-I-Islam. So therefore, we are very happy to introduce Anjuman-I-Islam, our parent body. And then coming back to this campus, from where you are now witnessing the short-term training program. This is a very interesting campus whose journey started, no doubt started in way back in 2002, a very small polytechnic, which was almost not known to anybody, got established on this huge land of over 10.5 acres. It was in 2002. Then till 2000, in 2002, it was only with 180 intake capacity. And in the very first batch, we got 38 students in Polytechnic. And it was just looking like this kind of building, a Polytechnic holding diploma programs. And till 2006, no much improvement. Overall strength of students were 255. So nothing great thing happened in 4 years after establishing this Polytechnic. But what happened after 2006 is really a benchmark achievement. So management body got changed and a new chairperson got appointed in the name of Mr. Burhan Haris. And after putting his experience in the field of HR and also IT training, so what he did is, in next 2 years only, this Polytechnic started attracting more students. The strength got enhanced from 255 to 400 plus. Then by 2017-18, the intake capacity of this Polytechnic became 720, 2,000 plus students, and relatively, it became the largest Polytechnic of Mumbai region. So with 2,000 plus students. So this was the journey till 2017-18 by Polytechnic alone. So when good things were happening from 2006 onwards, in 2010, Anjuman-I-Islam took a very, very important step for expanding this campus. And that has come in the way of developing a very interesting campus where multidisciplinary campus got established. So that is how here to our, if you see this to the left of this plot, we have got an integrated technical campus having 3 schools, and it was a milestone achievement in the entire history of Anjuman-I-Islam. School of Architecture, School of Pharmacy, and School of Engineering and Technology. So these 3 schools starting in 2011 was a benchmark for us. And not only starting, but in the next 10 years, it became a role model campus in the entire Anjuman-I-Islam. So in terms of its work culture, students' quality, the achievements, participation, and whatnot. And we are a very young institute, even getting NAC accreditation last year in 2019. And even NAC committee got surprised. So being such a young institute, so what was the need to go for NAC? So because we said we are focused on quality, so quality enhancement, quality assurance is our motto. So therefore we applied and we got NAC accredited also. So this was the campus which established in 2011. It was a jump start. And today, it is standing as a great, vibrant campus. And our goal is to really become a lighthouse for industry. And we have got all the plans in place. And this is how we are looking at to become a lighthouse for industry by establishing so many, centers and, center of excellence, around this campus. So all this is in pipeline. And in coming few years, we will emerge definitely as a lighthouse for industry with improved systems, and our people are very effective, very dedicated. So therefore, this dream will definitely come true. This is what our future vision is. So interestingly, I would like to particularly share with all participants that this kind of campus got established by the support of a single person, a single donor. So this huge campus having all these three institutions is not a joke, but Mr. Abdul Razak Kalsekar Sahab, who passed away in 2015, but he immediately accepted our proposal and funded the entire campus. It is to the tune of about 80 crores. And this person owe all that credit, and it became a dream come true for Anjuman-I-Islam. So such donors, such pious persons are there in the community who are taking lead to establish educational institutions of this caliber. So that is the reason why a pious person helped us. A campus got established, therefore good things are happening. And we are marching ahead to do good things. And this is how today integrated technical campus named Kalsekar Technical Campus having its UG courses. And we are the first to start PG program before our first batch of UG program was out. We have got PhD Research Center, and slowly we are also going to introduce PhD Research Center in other schools. So about 2,500 students are studying under this campus, and including Polytechnic, roughly about 4,500 students are a part of this campus exclusively. So that is the reason why it has become a role model campus for entire Anjuman-I-Islam. And we have got our core values, and these are not for the namesake, but we live up to these core values. So this campus is knit and work culture is developed by putting these 7 core values in place. And therefore, we have seen a vision to be the most sought after technical campus that others would wish to emulate. This is our vision, and our mission is to create exuberant technical professionals, be it architectural professionals, pharmaceutical professionals, or engineering professionals. So this is our mission of the campus. And this campus has got multidisciplinary activities. We gel with each other, and very interesting results are coming. So this is our vision, and we have adopted outcome-based education model where we want to ensure that 12 graduate attributes that are expected to be there in all technical graduates. So those we are looking at, and these requires implementation of outcome-based education model, and that is how we are marching ahead. And thanks to School of Pharmacy, they are very innovative. Though it all started as an MBA activity for engineering, but pharmacy took the lead, and they are doing excellent in implementing this outcome-based education model under the leadership of Dr. Sharik Sayed and all vibrant faculty members of School of Pharmacy. Thanks to them to make this OBE is really functional at practical level. And we ensure that our students are not only gaining their knowledge, but also we develop a lot of abilities and qualities that are required to be called as exuberant technical professionals. So these 3 pillars are strategic pillars of Anjuman-I-Islam's Kalseko Technical Campus. So therefore, we are doing well, and this is very briefly, introduction of our campus. And this campus is being taken care by these four, people. Of course, notionally, these four members, starting from our president, Dr. Zahir Kazi, who empowered all of us to do a good work for this campus, and our General Secretary, Mr. J.R. Sheikh, who is immediate in charge of our board. He ensures that we all are kept engaged in the right direction. And our Executive Chairperson, Mr. Burhan Haris, he enabled all of us to see that good activities are being conducted. And me, along with my all deans, HODs, the entire fraternity of faculty members, we are in the business of educating our students, and this is a student-centric campus. So with this brief introduction, let me take my seat. And now basically we are here to learn over this 1 week and weekend, 1 week, and even in the evening time, with all fresh mind. I wish you gain the requisite knowledge, that knowledge which will be useful for all of you. And therefore it is rightly said that we must know enough to know that we do not know. If this is our motto, then we keep on knowing more and more, and we keep on converting that knowledge into wisdom. And with that wish, I welcome once again all participants to this short-term training program. Welcome all. Thank you very much.
Thank you, sir, for introducing our parent body Anjuman-I-Islam and also our technical campus. Now, before starting actual session, now I introduce opening speaker of today's session, Dr. Muhammad Farooq Sheikh. Dr. Muhammad Farooq Sheikh is a senior lecturer at Monash University, Malaysia, and leader. Hello, am I audible?
Yes, yes, you are audible.
Okay, I'm sorry, there was some disconnect in between. Okay, so Dr. Muhammad Farooq Sheikh is a senior lecturer at Monash University, Malaysia, and leader of neuropharmacology research strength in Zebrafish School of Medicine and Health Sciences. He has completed his PhD in pharmacology from the Institute of Chemical Technology, University of Mumbai in 2012. He is an expert neuropharmacologist and interested in both drug discovery and understanding the disease mechanism. His team at Monash University, Malaysia, has developed a zebrafish model to study the comorbidities of epilepsy. This newly developed model using zebrafish will not only allow scientists to understand the mechanism of the disease, but also help in developing safer drugs. His team is also working on unrevealing the molecular mechanism involved in disease progression, especially the role of inflammatory biomarkers in epilepsy and other brain disorders. He was also awarded the IBRO APRC Fellowship to continue his postdoctorate studies at School of Biomedical Sciences, University of Queensland. He is a recipient of Monash PVC Award for Excellence in Research and Malaysian Society of Neuroscience Best Publication Award in 2017. Thanks, Dr. Muhammad Farooq Sheikh, for accepting our invitation and joining us. Meanwhile, before our resource person take up this session and share the slide, let me give you a few housekeeping instructions. To ensure learning and participants' engagement, I request our all delegates to actively attend today's session. Also, at the bottom of your screen, there is chat option. So for any query or questions if you have, then you please use the same. We will take up this question at the end of the session. Now I invite Dr. Muhammad Farooq Sheikh to start with his session on innovative disease model, a case study for zebrafish. Dr. Farooq, it is over to you.
Thank you, Professor Abu Sufyan, for your kind introduction. Very good evening to everyone back home. So very good evening to everyone. I hope everyone is safe and in pink of their health. Let me share my slide and then we can start. Yeah, I hope you can see my slides.
Yes, we can see.
Okay. All right. Thank you so much.
Yes.
Yeah. Thank you so much, Professor Abu Sufyan. Thank you so much, Dr. Sharik and Dr. Abdul Razak. I think this is a great opportunity to share my experience and the research we are doing here. But before that, I really would like to congratulate all the participants for making this happen. I think without participation, it's not possible to bring uh, the the workshop like this to a reality. And at the same time, I can completely see here, I have my very uh, precious and old friends here and my UDCT mates there. I can see Dr. Arundhati Abhyankar Lele. I can see Dr. Clara. I can see my old friend Dr. Amjad, Dr. Vaishali. Thank you so much. These are the people from whom actually I have learned a lot of things, and we had shared a lot of things in UDCT when I was doing my PhD. So thank you so much for coming, guys. So let me start with my presentation, and this is again a great opportunity to share what we are doing at Monash. Monash being the top 100 and world's top universities, among top universities. And I feel very fortunate and lucky actually to be part of this university. And I will say I have no regret in saying that Monash actually helped me to shape my career. Monash helped me to do research. Monash helped me to actually do teaching. So because if you look at the next qualification I have, which is GCHE, actually is a graduate certificate in higher education. So Monash uh, helped me to do a course at Monash. So I'm also a Monash alumni now, other than UDCT alumni, where actually I learned how actually we teach higher education or how we teach tertiary education. So this is where both research and teaching comes hand in hand. Uh, I really want to quote the Monash tagline, which is Ancora Imparo. That means I'm still learning, and this is very true. I'm still learning. There is no such a day where we don't learn. It's not part of our life. So learning is continued till we die. So let me go to the topic, and I'll spend some time on it to explain and discuss more. And I'm more than happy to answer whatever doubts or question you have in your mind. So don't hesitate to unmute and ask, or you can, if you are shy, you just put in the chat box. I'll be more than happy to actually address all those questions. So the title for today is Innovative Disease Model, and I'll be taking more, through you guys to you guys to the Zebrafish model, where we have done some research. Also, some of the important things why Zebrafish. A lot of people are still maybe in confusion, like, yeah, we are talking about translational and human research, and why and where and how Zebrafish actually play an important role. Just in a nutshell, I'm also leading Neuropharmacology Research Strength, which is a group of 15 faculty members with a number of research students. And I'm also head of my own lab, which is Neuropharmacology Research Lab, where I have one postdoc and about 10 PhD students doing their research. Then we have some masters as well as honors students. I have a couple of RAs who are also working and helping with our research. So I'm really thankful to all of them for the research what we are trying to do at Monash. So this is a quick outline of what I'm going to talk about. Translational research, a brief introduction I'll be giving you guys. Then drug development timeline, which most of you are aware, but we'll still go through. The animal models in drug discovery, zebrafish, some examples of epilepsy, which is something which is close to my heart. I think Clara and Arundhati will second that. Then I will talk about some of the advantages, what is screening, what are the models, and what are the success story from zebrafish models. So this is where I will be taking you guys for the next 45 minutes. So if you look at the translational research as such, it is a bidirectional process that involves multidisciplinary integration among basic and clinical sciences. So it's a connection between basic science and the clinical science if you talk about from the health drug research point of view. It could also apply to practice, population, and policy if it is the research is related to those areas. But here, I think most of you guys are from the biomedical science or pharmaceutical research background. So I will say translational from basic to clinical or from lab to the clinic. This is where the translational research is very, very important. And I think this is, we all as a researcher need to appreciate if as a researcher, we do not know the translational value of our research, then it probably we end up with either a thesis, if it is a student project, or it will end as a research publication. And that's all. The story ends there. And that means the research we have done, the time, money, efforts, uh, animals, chemicals, which we have sacrificed for the project, it does not have any translational value, no translational value. And if you talk to an industry person, then you will say it's such a waste, you know. And I feel as a researcher, when we get research grants, which is actually the money from the taxpayers, we have some responsibility. Whatever research we do, it definitely done with a lot of ethics and integrity. But at the same time, we also have value for the money. That means there should be some translational output which can help the people, which should help the population, which should help the taxpayer. And this is where, you know, the improvement in the health, the improvement in the quality of life as an outcome of a translational research is very, very important. And I think this whole workshop is talking about in relation to translational research. If our research is actually not having any translational value, then it will be very difficult. And I'll tell you guys, that means the translational value will not be immediate. May not be direct. But whenever, even if you are doing a very specific part, you should be aware what is the translational value of this research. And keeping in that mind, if you progress your research, that will give you more motivation and encouragement actually to continue research. So if you look at the figure down here, the translational research basically connecting basic and clinical research. And this is few terms, which is basic research. We try to find out how does it work or whether it's working or not. That could be another question. Then translational value or translational research will tell you, can it be applied to people? Can it be translated? Can it be scale up or can be transformed from clinic from the lab to the clinic? And the clinical research will tell you what are the effects. Does it really work, and how well does it work? And if, let's say, from the drug development point of view, if we say that whether it's safe, efficacious, effective, really improve the quality of life of the individuals. And I'm not going to go in detail of this drug discovery timeline. I'm sure all of you are well aware of it. And those, just for the knowledge of those who are not really aware of drug discovery, we have, you know, preclinical research. Before that, we have drug research. Then we have, let me take my pointer. Yeah, then we have clinical trials, and everyone very much aware of the phases. Then the evaluation process and the phase 4 or pharmacovigilance studies. We all, I think most, you all will agree, like most of our academic based, or you can say lab based research actually fits in this domain, which is drug research. And somehow, if we compare here what industry classically or commonly do, which is they screen hundreds of thousands of compounds in one shot. Okay, so they do have a very high throughput, high throughput screening, you know, methodologies which actually help them to screen. But if you look at a specific PhD thesis, someone from you know, pharmacology or someone from medicinal chemistry, we are talking about a specific compound. And very common in India, we do plant-based or ethnopharmacology-based research, which is absolutely fine. But we are already very much biased towards that plant, towards that extract, towards that molecule, and we try to prove everything positive at the end of the day. But the reason why all those research just close in the thesis and publication and does not get translated into clinics. The reason we really don't do very thorough evaluation, which is basically required for drug to be successful and make it to the clinic or to the market. And what industry does, it's hundreds of thousands of compounds. Having said that, now the scenario is changing. Now we have new things are coming up, which are drug repurposing. Okay, we already have a number of FDA-approved drugs, and now the research is concentrated more on no need to really discovering new drugs, but at the same time, let's try what available FDA-approved drugs are there, and then let's repurpose it for other use. And there are a number of examples. I'm not going to go in detail, which actually are repurposed. Classically, if you look at the repurposed drug, you can take an example of aspirin. You can take an example of minoxidil. Aspirin was famously initially developed as an anti-inflammatory, but if you look at the most common use in recent days is basically used for as antiplatelet drug. And then moreover, if you look at the recent research on aspirin, they say it's very good in preeclampsia. So if a pregnant woman who do have chances of getting preeclampsia, that means high hypertension during pregnancy, if very low dose of aspirin is given for about 9 months, then the preeclampsia can be prevented. So there are every day we see the new uses of the old drugs. And then we have minoxidil, very classic example. It's a beta blocker antihypertensive drug, but when we see the side effect, which is treating alopecia or growing hair, scientists see the opportunity to convert that side effect into a specific use. And minoxidil now not really commonly used as an antihypertensive drug, but it is used more for the treatment of alopecia. So this is, when we pay very close attention to the research we are doing, not only looking at the positive side or expected outcome, but also looking at unexpected. And this is where the serendipity, serendipity is very important in research. I think most of the drugs discovered so far are actually discovered by serendipity and not by a systemic, research process. So serendipity, those who are not aware, is like you are looking for one thing, but you end up getting another thing. I'll give a simple example of, epilepsy drug, which is phenobarbital. And phenobarbital was well discovered for, as a sedative drug or for inducing sleep. And what happened, there was a young neurologist who was placed in the ward of epileptic patients. And then what happened, he observed like a lot of patients actually falling from the bed after having epileptic seizure attacks. And this guy was very new, very young neurologist. He got a night shift. He kind of completely annoyed, like, come on, one after the another, the patients are falling and I'm so frustrated. What should I do? What he did, he took phenobarbital, injected to everyone. He was expecting like this phenobarbital will put all of them on sleep and then he can also relax. And what happened, he found that actually all these patients whom he injected actually phenobarbital were completely seizure free for next 2 weeks. But as a scientist, as a keen observer, as a responsible, a researcher or a medical professional, he actually reported that event, that accident, and then the phenobarbital actually categorized as anti-epileptic drug. So this is a typical, I will say, story of a serendipity based discovery of anti-epileptic drug. So as what I'm trying to say is we always pay attention to the unexpected as well when we are performing any research. So we know the drug discovery process is lengthy, about 15 to 18 years, about 1 billion euro. And then at the end of the day, you probably get one drug which may be safe and effective. I'm not going to go in clinical trials and all, which I'm sure you guys are well aware of. So this is another important thing, which is target-based to phenotype-based, drug discovery. And when we say target-based, So we find out a specific target. For example, if you look at hypertension, we look at whether a new drug really target any of the known antihypertensive targets, or is there a new target which can actually prevent hypertension. So classically, or so far at present, I will say most of the industries actually following target-based drug discovery approach. Fortunately, last year I was managed to attend Novartis drug discovery program, which is basically done by Novartis Institute of Biomedical Research based in USA. But with Monash, we have a collaboration that every year for next few years, they will run this drug discovery and development program in a fast track manner on our Monash Melbourne campus. And I was very surprised when the presenter actually said to us, like, for experimental or for drug discovery, you basically need targets, and you, as FDA, also do not want animal data. Okay, I'm talking about in general, but there are certain exceptions where you need animal data for the pharmacological efficacy results. But mostly the FDA want target-based drug discovery data, as well as they want the toxicity data, which is definitely where animals are involved. So I'm not saying that means like we do not need to do animal work. Yes, there are certain diseases which cannot be mimicked in non-animal based experiments. For example, epilepsy. We do not have a cell line based epilepsy screening. Even it is there, it is not acceptable by the anti-convulsant drug discovery program. So those cases are there. But there are another method which is, so I was saying, so most of the drugs are developed with the target-based screening. And then we have a phenotype-based screening. And this is where example of, epilepsy is there. Like you can really see a phenotype, because seizures actually a phenotype of epilepsy. So epilepsy is a root cause or the disease, but seizures are actually the phenotype of epilepsy. This is one of the example I'm trying to explain you guys as a phenotype-based drug discovery. And the screening is done based on the phenotype, whether a drug can stop the seizures, and then the heats are taken to the target deconvolution. That means after that, they will again looking at if it is showing anti-convulsion or anti-seizure efficacy or effect, then what are the targets which actually leads to this effect. So coming to the animals which are actually used in this drug discovery approach, and we have a number of animals, okay, right from a very old one like the cats and dogs, chicks, primates, and our own very famous one or very commonly used rodents, especially rats and mice. Other than that, we have rabbit, guinea pig, and others. But if you look at right from 19th century, which is, different models are used. And if you look at for the CNS related diseases, we have dogs, we have monkeys, we have fishes, and some of the lower vertebrates, which are having a somewhat similar, I will say, structure and function as compared to the human. And that if you look at, we have high throughput model coming or discovered around 1940, where there are a lot of fish models. Rodents actually came into picture even in early 19th century, but it got more famous during 1940s and during that period. Then also some primates were also get popularity in that time. Then if you look at 1980, there were more discovery on the cell-based screening, cell-based assay, and which is very commonly and widely used throughout the world these days. And then, OECD coming in picture, animal ethics coming into picture, and then they put more focus on 3R principle, which is refining, replacing, and reducing animal use in the drug discovery. So this is, the timeline of animal models, you can say. And then if you look at the up there, there are, there are a lot of phenotypic complexity in different animals. And on that basis, basically they are selected in different disease condition. And then you have factors where which are actually very similar into different other aspects, which is like genetics, model translatability, ease of experimentation, cost, and ethical concern. And this is where we moved from the higher vertebrate, which is like dogs, primates, chicks, to the lower one, where we are from rodents. Now we are moving to the more non-mammalian models like zebrafish, worms, Drosophila, and other lower species. So this is a brief outline of what is the timeline of these animals which are used in drug discovery research. And coming to the the innovative models or new models, we'll say we will look at it is coming with the yeast, then worms. Okay, C. elegans are what's very common ones. Then fruit flies with the Drosophila, then zebrafish and other fishes, but zebrafish having more advantage, and we're going to talk more about it. These all mimicking the human models of different diseases. So if you look at the mammals in the research, and if you look at the breakdown, which was early, this report is of 2010 from US FDA. Then if you look at that, major part of this pie is actually captured by rodents, specifically mice and rats. And to a minor extent, all the other, right from guinea pig, rabbits, hamsters, cat, dogs, and fishes, including, were having a very small part of this pie. But if you look at in today's scenario, the pie is taken much by fish, and it is eaten up some of the pie of the rats. But again, mice being very popular due to various reasons. But I think slowly this again will be captured by the fruit fly and the fish in the future, time. So coming to the topic we're going to talk more in detail today is the zebrafish. And then I'm going to take you guys through the different examples of zebrafish. But before that, I want to talk to you why zebrafish, as I mentioned in the outline. And if you look at the comparison between the human major organs and zebrafish major organs, it does have about a lot of similarity among these major organs. If you look at the liver, yes, zebrafish do have a defined liver. Pancreas, that's wonderful. That makes us to think, yes, it can be used in diabetes research. Then it does have a gallbladder, which is not present in rodent models. It has a very small but defined circulatory system. That's nice. If you want to even study stroke models, that can be done because we have a very small circulation system, and everything is very transparent and can be visible and see without having very strong imaging or very, I will say, high-end technologies. What about digestive system? Yes, zebrafish do have a digestive system, intestine including. So if you want to study some of the digestive tract related diseases, that can be very much done here. About the lungs, it does not have lungs, but it has gills, which play a kind of similar role. Then the important one, which I'm interested in, is central nervous system. They do have a very defined brain and brain structure, but at a smaller scale, that simplify as a neuroscientist or a neuropharmacologist to do a lot of or to study a lot of things in that small brain. If you look at the genes, about 70% of the genes from human and zebrafish actually are similar, and about 84%, to be very precise, the genes which are in human are also present in zebrafish, which are disease-associated human genes. So I think that's fair enough, actually, to convince us like zebrafish is an ideal model to be a disease model, and then we can study various diseases using zebrafish model. So moving on, why again zebrafish? So other than the modeling we are talking about, there are various reasons. And one of my favorite, this figure is from one of our paper published in Frontiers in Pharmacology. And one of the reasons why I feel zebrafish is much more better than in vitro or cell-based method, because you just take an example of a CNS drug, okay? A drug, it shows a very good activity in cell culture. For example, if you're looking at neuroprotection, or if you're looking at anti-neurodegenerative or anti-Alzheimer's effect, you may get very good effect in neuroblastoma or SY55 cells, but a lot of drugs fails when it was tried in rodents. The reason why in vitro model does not represent blood-brain barrier, which is the main, I will say, the rate determining step when it comes to CNS drugs. If drug is showing effect in cell culture, but it does not cross blood-brain barrier, that drug is completely a failure. Okay, so in comparison to the in vitro model, I feel the zebrafish model, being very small, being very economical, it gives the opportunity actually to screen the drug, in a dynamic system, in a zebrafish which has all the major organs which I spoke just in the previous slides. And this is really fantastic and give us an opportunity actually to screen drug in zebrafish rather than in vitro model. Having said that, that doesn't mean in vitro system are not good. Definitely in vitro system does have its own advantages in terms of doing rapid screening of various drugs. And I think for cancer cell lines, for cancer work, in vitro work, actually the gold standard, which helps do a lot of things using the in vitro screening model. The other reason I was talking about zebrafish, if you look at the rodent models, rodent is a nocturnal animal. Okay, so if you want to do a sleep research using zebrafish, I think it is difficult to do in rodents, although people have reported and published, and so many things are there. But rodents are nocturnal. They are very active in the dark, in the night, and they are sleeping in the day, which is very opposite what human does. Whereas zebrafish being diurnal, it has same routine of sleep wake cycle as humans. So that's people may ask whether zebrafish really sleeps. Yes, researchers has defined how zebrafish sleeps. Some says it sleeps in the bottom of the tank. Some says it's sleeps at the top of the tank. And if it is mobile more than 6 seconds, that means it is sleeping. And researchers have done a lot of studies using the electroencephalographic EEG recording, whether to see whether they are in a sleeping state or not. So there is no doubt zebrafish is a good model to study sleep research as well. What about BBB permeability? I said it has a very stringent, similar blood-brain barrier as that of humans. So no doubt it can be used. Zebrafish allows to do gene expression study, and I think, as I said earlier, we have looked at the 70% similarity to the human genes, 84% similarity to the human disease genes. It's absolutely excellent model to do gene expression study to understand the mechanism, how your drug, which you are testing, is working. It is really, cost-effective model, highly cost-effective model, very cheap. You can do a lot of research in a small amount of money, and so that's the best part. Low volume of drug is required. Okay, same as in vitro model. In vitro model are famous because you want to do high throughput screening, and you need a very small amount of drug. So this is same advantage, go hand in hand with the zebrafish model as well. It is rapid, as good as the in vitro model. I will say it's faster than that, and also reliable. As I mentioned earlier, it is a living organism. The last part, we will think like, oh, can we do Pkpd studies in zebrafish? The answer is yes. Efficacy, no doubt about that. PD is no doubt about that. But whether PK pharmacokinetic can be done in zebrafish, researchers have developed different models, different chips actually to take a very small, which is microliter amount of blood from zebrafish, and then the PK studies can be done. So if you look at the why zebrafish, this is one from the Nature paper, and it says it conserves the targets, it conserves the metabolism, same as the human, it conserves the physiology as of human, and it conserves the pharmacology as well. So there are a lot of reasons which are conserved in along this species, and also we can find that in zebrafish that convince us more. Yes, zebrafish is the model which can go further. Another great advantage of zebrafish. Zebrafish actually develop within the embryo. That means outside the body. Okay, so the fishes, the female fishes lay eggs, and then the eggs actually develop outside the body. So this is very opportunistic thing, because this allows researchers to study developmental disorder using zebrafish embryo. So it is being very transparent until the larvae level. That makes the life of researcher very easy. That means with a simple compound microscope, you will be able to observe the whole development. In fact, if you Google, you will find videos or GIF where you can observe the whole developmental cycle of the zebrafish within few seconds or within few minutes. That's wonderful. So this allows us actually to screen drug. If you want to do, developmental toxicity, very much can be done in zebrafish within less than 24 hours or 72 hours or 96 hours based on the OECD guideline you are referring to. And then the next stage, which is the larval stage, within 96 hours, actually zebrafish completely developed. They have all the organs, the brain is completely developed, everything is fine. At the same time, the advantage is this zebrafish is completely transparent. Okay, so if you lose again simple microscope, you can see heart beating, you can see the blood flowing, and you can completely see the transparent zebrafish. So that's another opportunity. If you want to test your drug using this larval model, that's another stage of zebrafish where you can do the testing. So I talk about embryo stage, I talk about larval stage, and then you have adult zebrafish, which is actually the full package, different disease models which are done on zebrafish model. Whatever disease you name, if you Google or you are interested in, you will find zebrafish model, and that can very much be done using the zebrafish model. So again, I'm trying to convince you guys, like this looks like an excellent model. When you compare to rodents, your pup's gonna take some weeks, then it become adult, it's gonna take again some weeks, and here we are talking about within hours. So we are facilitating the drug discovery process using the zebrafish model, which is gonna be very, very rapid. So this also gives an opportunity to study developmental issue, as I mentioned, developmental disorder, genetic mutations. Okay, in this embryo state, actually you can knock in or knock out genes you are interested. You can use morpholinos. You can insert genes, and that makes a specific genetic disorder model. If you're trying to create, it makes your life easy in zebrafish model compared to rodent models in terms of cost, in terms of timeline, in terms of the reproducibility. This is another the diagram, Venn diagram, which is showing when we compare not only with the humans but also with the other models. Zebrafish do have a lot of similarities, so we did talk about this earlier, so I'm not going to go in detail again. So I did spoke about at different stages, you have embryos, you have larvae, you have adult zebrafish, and this gives you an opportunity to directly test compound, to modify using the CRISPR technique or other morpholino technique, and then you can do high throughput screening. For a larvae or for an embryo, you just need the same 96 well plate, which we do normally for the cell culture, and the larvae can actually fit in well in a single well, and then you can have 96 well for 96 larvae, and you can do the rapid screening same as you do in vitro. But you will be able to record the behavior. You're able to see the behavior abnormality. If you're doing toxicity study, It's an excellent model. You can see the toxicity in the larval as well as embryo, as well as the adult model of zebrafish. You can study the molecular level, you can study at cellular level, you can study the behavior level, and we will talk more about it in the coming slides. So the behavior I was talking about, and if this is just an example of, various behaviors like anxiety, shouting behavior, social preference, learning and memory behavior, and, other important behavior like fear conditioning or, aggressive behavior. Zebrafish already have a well-established models. Researchers have kind of very well established some of these models and validated these models. And on the left side, if you see, there are comparison with the available rodent models. So if you talk about the light and dark model, which normally used in learning and memory testing in rodents, actually very much available in the zebrafish as well. If you talk about the open field test, which can very much done in the novel anxiety test in zebrafish, and then you also have, fear conditioning or learning and memory, how we have plus maze or T maze for rodents. Similarly, we have this for zebrafish as well. So we did talk about the genetics, we did talk about molecular mechanism. Now I'm talking about even behavioral aspects, and a great detail, very minute detail of those behaviors can be well studied in zebrafish. And this is a typical timeline, and you can actually try to find out the area of research or disease model which you are interested and trying to do. But if you look at right from 1960, where zebrafish was discovered in the rivers of Ganges in 1822, and this is very much our Indian origin zebrafish. And if you see now for research, it is used more by foreign researchers rather than Indian researchers. But now, if you see from the Ganges, now it is available all around the world, and people all around the world have developed different, libraries of different strains of zebrafish. Like if you talk about Japan, we have a Riken type of zebrafish. That means they have modified there, and they have streamlined their strain. If you talk about Singapore, then we have an AB strain which is present there, and various other strains which are available for zebrafish from the wild type of zebrafish concern. If you talk about genetic models, yes, people have created a number of genetic models, and I'll be talking a bit about some of the genetic models which we have used for our epilepsy research, specifically the gamma aminobutyric acid, mutants, as well as the sodium channel 1 mutants. We will talk about that in a minute. So if you look at the different, conditions or different conditions which you can actually see your area of interest, where the zebrafish can be used. But in general, if I say, and if you read a different disease condition mentioned here, we see CNS drugs or CNS diseases are actually very commonly used because of a very well-defined brain in the zebrafish. And in 2017, actually, we have contributed this by developing a model for epilepsy-induced cognitive dysfunction model, which was published in the Frontiers in Pharmacology, and we have received very high number of citations for that paper. So again, as I mentioned earlier, now we are talking about the toxicology where the zebrafish is in use. And if you look at organ-specific toxicity, behavioral toxicity, systemic phenotyping, metabolomics, and toxicity reporter lines, all can be done using zebrafish. Why I put this slide of toxicity? Because if you talk about the drug discovery process, overall toxicology is of great value when it comes to FDA reports. Unfortunately, FDA haven't recognized zebrafish toxicity as one of the criteria to approve a drug application. What does it mean? That means still FDA want the rodents and the mammalian animal data for safety and efficacy. But again, before you invest your time, money, and energy in doing that large preclinical toxicity trials, I will suggest Zebrafish actually give you phase 0 a kind of opportunity to do those toxicity studies, because we know a lot of drugs, because of toxicity, they fail in the later stage. So I think this is a good opportunity to prevent that to happen. If we talk about COVID-19 research, that's the hot cake for the nowadays, and people have actually developed this model by infecting zebrafish with COVID virus, and then that become a high throughput screening model. If you want to do drug repurposing, if you want to reset new molecules or entity, definitely then that is done using the zebrafish model as well. And this is typically how they did. There's a timeline. If anyone interested, it is there. I took it from one of the published papers. So they are talking about, yes, coronavirus to the zebrafish larvae. Again, larvae being very popular because it gives opportunity to do high throughput screening, and then incubation, your drug treatment, and then assessment using the bioimager in this case. But you can always modify and do whatever you are interested. So I'm going to go to my topic as a case study, what we have used zebrafish in epilepsy. As you guys are aware, epilepsy is a serious neurological disorder, and which is related with the muscle jerk and convulsions, and it is also referred as an electrical storm in the brain. So I'm not going to go very much detail of what epilepsy is, but some of the signs and symptoms of epileptic seizures. Many of you guys are aware, or you probably knowing people who actually suffering from epileptic seizures. They have sudden fall. This is very common if they are having tonic-clonic type of seizures, or aura confusion if they are in absent seizures. Then staring, loss of consciousness, strange sensations, strange smells, emotions, uncontrolled jerky movements. These are very much. Why I'm saying that? Because we would like to mimic those kind of some of the symptoms in the zebrafish model or in the model you are planning to use. So to be very precise, what we are looking at molecular mechanisms. We want to look at neurotransmitter. We want to look at GABA. We want to look at glutamate. We want to look at sodium channels. We would like to look at gene expressions or genes which are involved. We want to look at the electroencephalography, which is a gold standard for epilepsy diagnosis. And then we want to look at the genetic models, also the behavioral outcome, which is tonic-clonic phase of this disease. So why these models? Because we want to use the model that recapitulate some of or all observed pathologies of the disease. This is what I said in the previous slide. And if we could study both genetic and the phenotypic basis of the disease outcomes. So we need a system in which we can test the potential or the potential drug candidates. So for our study, we found, yes, this is perfect. And again, we try to summarize the reasons for that. So zebrafish, we can study efficacy or pharmacology of it, which is very much required in epilepsy research. Then looking at the behavior, we wanted to study the electrophysiology, whether there is it is just a muscle movement by which we see seizures, or it's a real epileptic seizures that can be seen using the action potential or EEG recording. If you look at the genetic models, you can have gene editing or morphine injection. So we found, yes, zebrafish larvae, adult zebrafish, both are very interesting model to that. You can actually do ictal recording or action potential from zebrafish. And if you see, this is an example of a normal EEG recording or action potential rather in zebrafish case. And then when you have onset of seizures, it goes increase amplitude and frequency. And when there is a complete seizure, high frequency of this amplitude and frequency, and in conic. So what I'm trying to say, there is increase in the EEG frequency. And then people have developed, actually, this is by a Korean group, which they have developed a chip which can be implanted on the zebrafish head, and that give you a non-wired EEG recording. This is not commonly available to everyone, and somehow this chip they have developed is using the gold, as a transmitter, and definitely it's gonna become a bit expensive. At the same time, that become more accurate, and actually that is in use. This is something which is there from NIH, the National Institute of Health, for their anti-convergence screening program. And if you typically see what they are interested in, initially, if you are looking at anti-convergent drug development, they want a maximum electroshock model, or they want pentylene tetrazone induced epilepsy. I think many people are aware of that chemicals, which actually used to induce epileptic seizures. I will say rather seizures, non-epileptic seizures, induce the phenotype, which is seizures. And then if drugs are effective, then it will go to the further process and looking at the different other models of epileptic seizures. And then you have two different models. So you have acquired epilepsy. That means people who get a head injury, they get trauma, brain tumor, stroke, Alzheimer's disease, or other neurodegenerative diseases. And because of that, they get acquired epilepsy. It's not genetic, it's acquired. We call it acquired epilepsy. So that models can be very much mimicked by the chemical induced or hypothermia induced models. And those who have a genetic problem, mutation of SCN1, mutation of GABRA1, mutation of gamma2, or mutation of other genes which are responsible for epileptic disorders, then that models are actually genetic models. And the population of genetic models are actually just 5%, whereas 90 to 94% of the cases are acquired epilepsy cases. So that both can be actually very much done in the zebrafish model. So this is example where you want to inject morpholino to bring to or to make a mutant phenotypes in zebrafish, looking at very specific behavior or very specific aspect of a disease in a genetic screening. That can be very much done. And this is one of the paper I was talking about earlier, which is zebrafish as a model for epilepsy-induced cognitive dysfunction. We able to study pharmacological, biochemical, and behavior aspects of that disease. It was done by one of my PhD students. Now he is a postdoc in University of Montreal in Canada, Uday. He's also from Mumbai. So what we have done in this model, we have used pentylene tetrazole 170 mg per kg, which we have optimized in our lab using intraperitoneal injection, because we want to make sure the quantity we are injecting in these fishes actually are more accurate. There is another method by which you can actually induce seizures by dissolving the pentylene tetrazole into the water directly. But the problem is with that, you need to have more quantity or large quantity of pentylene tetrazole. At the same time, if you are also using that method to use as a drug treatment or the potential candidate you are trying to evaluate, then you need to have a larger volume or amount of that drugs. Then after injecting PTZ, we try to record the video using a video cam. And then we have our software. It's called Smart Software from PanLab Harvard apparatus. And then that make us to evaluate the tracking pattern, the seizure score, onset latency, as well as locomotor parameters, which are common parameters used in seizure assessment, and that we actually do with this model. And I'm gonna I hope this will work. Unfortunately, this video is not working. Never mind. So this was a video which is showing how epileptic seizures looks like, but just sorry for that. It looks like it's not working. Oh yeah, sorry. Okay, so this is the video which demonstrate when we use intraperitoneal injection of epileptic and pro-convulsant drug pentylene tetrazole. So these are different scores which able to evaluate using zebrafish model. So in the beginning, it will swim or do a short swimming at the bottom of the tank, which you can see. This is not normal. This is not a normal swimming in zebrafish. And then a second score is increase in the swimming activity. So you can see the swimming activity is kind of increased in the zebrafish. And the third parameter we were looking at is the burst swimming, okay? Moving left and right and erratic movements, which you just saw, or you can see on your screen. And then we move to the next stage of this zebrafish seizure behavior. Basically, this model is very much established by Mussolini et al. In 2013, but we try to validate in our lab. And the fourth score is secondary circular movement. And if you look at the tracking pattern, you will be able to see to a great extent how the circular movement looks like. But if you can still observe some of the behaviors which are the circular movements. And then the fifth one is a clonic seizures, which is whole body rhythm and muscular movement, which will be seen now. You see, that's pretty bad seizures, which we actually try. We see some of the people with epilepsy. They show the whole conversions of the body, and then in the end, which is sitting on the bottom of the tank, which is similar to the stupor phase, which actually seen in patient after epilepsy attack. They will sit quietly or they are unconscious. So this is how we have actually draw that, like short swimming, increased activity, and all those parameters what you have just witnessed in the video, we have put into the figure as well. And this paper, this book chapter is due in the Springer Nature publication with neuropaths as a book chapter. I'm happy to share once I get. So what we have observed from that, if you do the video recording and then software analysis for the tracking of the swimming activity of these control fishes, it's absolutely all over the tank. This is normal swimming. But if you look at the PTZ, you can see the burst in the swimming, and the activity is absolutely abnormal, which is an epileptic seizure kind of activity. So these are some of the results. I'm not going to go in detail, but if you look at, we're able to see there is decrease in seizure score once we treated with the anti-epileptic drug, and then onset time was also delayed when we look at the treatment one. And these are the different patterns when we look at PTC control, and then you see diazepam and other standard anti-epileptic drug actually try to bring it back to the normal as same as the control groups. And this is some of the learning and memory behavior which we have done in that paper. And this shows, how fishes actually, when we expose at 0 hours, 3 hours, and 24 hours, this 0 and 3 hours are basically the training or learning, and then 24 hours is a memory. You see the 24 hours fish actually get into the right compartment within 9.5 seconds. Whereas the first day exposure, the fish is still roaming around in the the main arm as well as on the left arm, and it takes a longer time, which is about 32 seconds. Whereas the second exposure actually took about 16 seconds. So that's clearly indicate a learning and memory behavior. And we found that the fishes which we induce seizures, or you're looking at any other inducing agent which impair learning and memory, then you can see a clear difference, same as what you see in rodent models. So when we track, you can clearly see this is actually going round and round, and then it takes more time to enter, whereas this one just quickly enter to the goal chamber. So TMACE, this is something which we have used, and we have calculated the inflection ratio, which is the transfer latency of different time points, and then inflection ratio is much better compared to just the transfer latency. If you're interested, we can talk later more about it. So these are some results from the TMACE case. And as I showed in previous figures, we checked with all the anti-epileptic drugs. Interestingly, what we found is, anti-epileptic drugs, they were good in preventing seizures and improving the seizure effect. But when we look at the learning and memory, they have negative impact on the learning and memory. That means the patient who are taking anti-epileptic drugs, they get probably prevented from their epileptic seizures, but those drugs actually have negative impact on their learning and memory behaviors. And we know there are a lot of serious side effects associated with anti-epileptic drugs, which are available as of now. This is another model for chronic epilepsy we have developed using repetitive administration of pentylentretrazole and then single administration of phenic acid to develop chronic epilepsy model. This is again done by my PhD student Uday. And this is again a complicated learning and memory test, which is called 3-axis maze. And fishes are actually allowed, or they're asked to, explore using different compartments, which we have only one opening, which are chained at different position. And this is the last day, the 10th day after training. We found that the fishes which are done a lot of training for 10 days, they quickly go into the 18-second. And fishes which have memory impairment, either you use scopolamine or use epilepsy-related memory impairment, we found they are lost and they found difficulties actually to reach the deeper chamber. So that allows us to test our drugs on those models. And this is a typical tracking pattern. I'm not going to repeat. I think it's very clear. It get lost with the PTZ, and it was very straightforward in the vehicle control. So again, this screening of epilepsy can be done very much in the 96 well plates. And then you do a similar thing what you do in cell culture. You put the larvae, which is 6 days post-fertilization in each well, one, and then you induce seizures with 20 millimolar concentration of pentylentretrazole, and then observe under camera and record video for 10 minutes to look at the epileptic seizure behavior. This slide I dedicate to my, this is from my PhD student, Vanessa, and this is what she has done for. And this is the same thing we have also done using the genetic models with our collaborator in Center of Research, University of Montreal in Canada. They use this Denue vision apparatus to do the screening, and then same procedure, but these are the genetically modified or mutant fishes. Wanted to look at whether our compound really work in the genetic mutant model as well. So we check the maximum acceleration as an evaluation point. The same we have done for the SCN1 model as well. And then this is the slide from my another student, which is Brandon, who is doing a PhD right now in third year. We have actually screened our high throughput screening for some derivatives, what we have the novel derivatives. And then we check using the primary PTZ model. We got some hits from there. And then we check these hits in different genetic models. And then we came to this funnel, which is showing us like after this hit, we got few more, hits. And then we are actually at this stage where we have about 4 compounds which are very potential in genetic models of epilepsy, and hopefully we will able to translate into a bigger trial or in patients. So my student Brandon is working on that, and our collaborator Eric Samret, where actually Brandon get a chance to travel to Canada last year and do his work. Eric also helped us to do the imaging thing, whether it's really epileptic seizures, so look at the brain activity, or it just muscle activity. So this is something which is upcoming, but I shared you as an example where actually we can use Zebrafish as a screening model to develop new drugs. So this is some of the results from Vanessa's unpublished data. And then you can see the knockout model where you can see increase in acceleration, and some of the treatment actually try to prevent that or control those increasing movement because of the epileptic seizures. So what I'm trying to say that epilepsy model is actually gaining a lot of popularity, and it is actually working well in epilepsy as a case, and which allows us to identify epilepsy alleles, is give us opportunity to do some editing, do high throughput screening, do EEG confirmation, also the toxicity study. So this basically taking us towards the precision medicine. I'll also share an example. For example, if we know a patient is suffering from a specific genetic disorder, or that could be rare, if it is a genetic epilepsy, but it's a rare kind of genetic epilepsy, what we can do, we can actually mutate that specific gene in zebrafish, test the existing drug, which drug works for that particular specific genetic mutant zebrafish, and then translate back to this patient, which is having a specific. So we are moving towards the precision medicine using zebrafish model without going through any bottlenecks of drug discovery process, but it can be translated by doing drug repurposing using zebrafish models. So these are some of the success stories. Zebrafish actually earned their drug discovery stripes, and this is the research done by the Barabun lab in UCSF, University of California, San Francisco, and he's testing using this clemizole, which is an antihistamine drug, and when he did high throughput screening using SCN1 mutant model, which is a model for Dravet syndrome, he found this clemizole actually works well in Dravet syndrome model, and that data actually he able to convince FDA without going any further screening to directly test into humans. And this drug actually made a lot of progress in phase 1 and phase 2 trials. And so what I'm trying to say, zebrafish model to human without any other bottlenecks, actually we can see a real translational value, which is an example from the Barabun's lab in case of clemizole. That is really very encouraging, and a lot of people actually going into that direction. So this is the paper from again Barabun's group talking about clemizole, which basically modulates the serotonin signaling and suppress seizures in Dravet syndrome model. Dravet syndrome is a very devastating epilepsy syndrome, specifically in children which do have SCN1, which is sodium channel 1 mutation. And these kids does not really survive for long, and they every day they have number of seizure attacks. And this really, life-changing news for those parents who are having Dravet syndrome, epileptic, children, and clemizole in trial. A lot of them actually benefiting from that. So that's really a good news, which actually zebrafish helps to do or improve the quality of life of these children. So to end with that, I can say, this is the quote again from UCSF, that the zebrafish really earns its stripes in epilepsy translational research. With that, I would like to thank every one of you for your kind attention. And I would like to thank my whole group, some of the present as well as the alumni. And then I would like to thank the funders for the research, especially the Ministry of Science and Technology, Malaysia, and Ministry of Agriculture and Agro-Based Industry, MOA, Ministry of Higher Education for funding different projects. And my special thanks to the Center for Research in the University of Montreal, Prof. Pierre Drapeau, as well as Dr. Eric Sambrut, along with Uday and Brandon, who actually did a lot of work using a zebrafish model. So thank you so much for your attention. I'm more than happy to take questions. If you guys have any, you can either directly ask by unmuting me, or you can write in the chat. So over to you, Dr. Sufyan.
Well, thank you, Dr. Mohammad Farooq, for a very informative session on innovative disease model, a case study for zebrafish. You have given wonderful case study of phenobarbital, importance of zebrafish model for studying anticonvulsant, neuropharmacological, and other studies. You have also covered other model in line with 3R principle and comparison of zebrafish phenotyping with human model and many more. Now let's take a few questions. I think I can see there are few questions in the chat box.
Yeah. So I think where to start with.
Okay, you can also take up directly from there.
Okay, so zebrafish models are also supervised by CPC. Yes, zebrafish. If you're talking about adult zebrafish, you do need animal ethics approval, irrespective of whichever country you are and institutional body. But if you are talking about larval until 7 day post-fertilization or embryo. That means you do not need any ethics. That's international guideline. But if you are doing adult zebrafish, definitely you need animal ethics approval. I think that's what Mona Yogesh Agarwal mean by. What are different kind of disease are benefiting from zebrafish research? I think I have given very detailed explanation, but I share a very good example of epilepsy. It's really benefiting. But there are other models which are toxicity study made very easy using the zebrafish model. A lot of people actually using zebrafish for the cancer research because you can do the xenografting and all. A lot of genetic, overall the genetic models, zebrafish actually doing wonderful work. If you ask in my lab, we're also looking at the learning and memory other than epilepsy. We're also looking at Alzheimer's model using zebrafish model. We're also looking at neurotoxicity using zebrafish model. And recently with my postdoc and my new PhD student, we are working on traumatic brain injury model in zebrafish. So this is something, and also the post-traumatic epilepsy models, we are actually using zebrafish model. Somebody, Dr. Meenal asked, session should be weight of zebrafish to be used in the study. Yes, you always, because I think as a pharmacologist, I do believe we want dosing to be done based on the weight, and you can anesthetize, you can weigh the zebrafish and calculate your dose accordingly, and then you can inject either intraperitoneally or you dissolve in the water, or you can do oral dosing as well in zebrafish. There are established method for that. And to name, I think in Dr. Reddy's Institute in Hyderabad, Dr. Pushkar Kulkarni is someone, I will say, one of the pioneer who actually did a lot of work in zebrafish, so that his papers can also be referred. But I'm more than happy if anyone interested and would like to collaborate or need any help, I'm more than happy to help on that. Somebody, Sabah is asking on topics. I think, Sabah, that we need a separate discussion if you're interested, or you can get in touch with Dr. Abu Sufyan, because we are also in discussion on doing some of the collaborative work using zebrafish. You can also talk about other people from BCP. We have Dr. Clara, we have Dr. Vaishali, where we have discussed some zebrafish models. I think Dr. Vaishali is in particular interested in doing that. Mr. Sankar asking, can we check anti-cancer activity as that of MTD assays using zebrafish model? What are challenges associated with zebrafish model? Certainly, as I mentioned, yes, you can do that. But I will still say when it comes to the target, because when you look at the cancer cell, it's a specialized cell, and your target is just cells, and you do not have any problem of blood-brain barrier. If you do not have any other problem, I think because the MTD assay you're going to use is the human cancer cells. This is the closest we can go from the human condition. So I will still, I will still support the human cancer cells for anti-cancer work. But if you're looking at in vivo or tumor and all, then I will say you can look at the zebrafish model as well. What is the rational strategies of drug selection for drug repurposing? If you ask Jayashri, there is no, if I'm not mistaken, if Jayashri is a university alumni or it's a similar name, I'm not really sure, but I know one of the Jayashri. So I think drug repurposing is basically completely screening based. It's not really specific, but you can actually do some docking modeling before if you want to rationalize it. Otherwise, it's very much on the screening based. Dr. Surendra asked, BB work in zebrafish can be correlated to human BBB. Yes, absolutely, because it has a very similar structure, very strong, I will say, connection from the brain cell one to another, the barrier, the BBB, the blood-brain barrier, the endothelial cells, and the connections, the tight junction is absolutely similar to what the human blood-brain barrier is. So if you are looking at BBB, blood-brain barrier permeability, blood-brain barrier toxicity, you can still use zebrafish, that excellent model. Thank you guys for your comment. I think I'm
Thank
done
you.
with answering most of the questions, unless anyone has
I
specific
just,
question.
Dr. Farooq, I just have a last question. I think we're really short of time. Dr. Abdul Basit has joined in from West
Okay.
Coast, US. So just one last question. So with regards to your experience and your research around there, are there any documented uses of Zebrafish as such in, industrial drug development where, companies have reported that into their, clin farm summaries or other places where they have shown that we have used zebrafish, and in which therapeutic areas? I am sure most likely it would be more into neuropharmacology, mostly into Alzheimer's and stuff. So any
So Dr. Sharik,
information?
I think you asked a very important question, but I think your own experience with industry is very much interested in targets. And if I quote one of the statement from the Novartis director, he said, "We are not at all interested in animal models."
Right.
Okay, so because FDA doesn't want that data, why they should spend the time? But this is where the reason why the most of the discoveries are actually coming from academia and
Hmm.
not from industry.
Right. Right.
Okay, yeah. So I think this is the reason why
That is
when
true
we
to
are
a certain extent, but what FDA really these days is also using it. There's a critical path initiative where they are, looking at trying to bring innovation into the clinical trials aspect, and in innovation, as I mentioned earlier, into every aspect of it. So if
yeah, yeah.
it
So
is
so if you look
innovation
at
in
one of the
terms, in
one of
terms of
the
the models that we use to expedite our understanding of a disease, obviously, there is acceptance to that.
Yeah, I completely agree on that. And if you look at the one of the news on the FDA website, they clearly mention like zebrafish actually earns its splash. It's
Right. Right.
showing its splash on the drug discovery research. So FDA is accepting zebrafish, but again, to be very precise, we are talking about genetic models where
Hmm.
zebrafish model has more acceptance compared to the acquired kind of diseases or condition. A
Okay.
specific example,
Okay.
epilepsy. If you talk about acquired model, if you're using zebrafish model, very unlikely it will convince the reviewers. But
Mhm.
if you are using for genetic epilepsy, and I gave example of clemizole, which is a very we'll say live example of translational research using zebrafish as a model.
Right, right. Makes sense. Makes sense.
Thank you
Great.
so much.
Thank you so much, Dr. Farooq. In the interest of time, I think if there are any residual questions, we can take at the end. Dr. Abdul Basit has joined in. So, Professor Abu Sufyan, over to you. Thank you once again, Dr. Farooq.
Awesome. Thank you so much.
Okay, sir. So once again, I thanks to Dr. Mohammad Farooq for highly informative and wonderful session, and also taking up our participant questions and clarifying their all doubts. Thank you, sir. Going ahead for a day one session 2, we have with us Dr. Abdul Basit. Dr. Abdul Basit is an assistant research professor in Department of Pharmaceutical Sciences, College of Pharmacy and Pharmaceutical Sciences, Washington State University, Spokane. He has worked as senior fellow and technical director for quantitative proteomics and metabolomics as at Prasad Lab in the Department of Pharmaceutics, School of Pharmacy, University of Washington, Seattle. He also has working experience with in vitro DMPK at Sai Life Science and Vanta Bioscience in India. Dr. Basit has published more than 30 peer review articles, more than 40 conference abstracts, and filed 2 patents. His current research focus deal with biomarker discovery for drug metabolizing enzyme and transporters using mass spectrophotometry for the prediction of drug-drug interaction, adverse, adverse therapeutic events associated with narrow therapeutic window drugs, and prediction of precision dosing based on biomarker. Thank you, Dr. Abdul Basit, for joining us. Maybe it is very early evening for you. I think it is 5 a.m. there in the US. Now, with this brief introduction, I invite Dr. Abdul Basit to start with his session on drug transporters, case for inter-individual variability in drug exposure. Dr. Abdul Basit, it is over to you.
Thank you, Dr. Abu Sufyan, for a nice introduction. Can you hear me?
Yes, you are audible, sir.
Okay, yeah, thank you. So let me share my slides. Okay, can you see the slides?
Yes, it is visible.
Okay. Yeah.
You can make it full screen.
Yeah.
All right.
Okay. Good evening, everybody. And first, I would like to thank the organizing committee for the invitation to present at this workshop. And today I will discuss about the drug transporters and how its variability across across different species and in human itself affects drug development. So this is the outline of the today's presentation. So I'll give in a couple initial slides. I will talk about the introduction to transporters and how, these transporters are classified and where they are localized in the human body. Then in some of the slides, I'll be talking about like why we should study these transporters and with the examples related to the clinical significance. Then we'll talk about the inter-individual variability in humans and some variability across different species and how these are, translatable into the human. And at the end, we'll discuss a couple of applications in drug development. So transporters, these are the membrane-associated proteins that play an important role in ADME process, like absorption, distribution, metabolism, and excretion of the drugs. And many of these transporters have been cloned and characterized and in human genome. And in drug development, particular attention have been paid to the transporter expressed in the epithelia of intestine and the liver, kidney, and blood-brain barrier. There are numerous studies that have suggested that the transporters play a part in vivo in drug disposition, therapeutic efficacy, and adverse drug reactions. And the in vivo role of this transporter is demonstrated in several animal species, including knockout mouse and by the loss of function genetic variants in human. So there is a lot of progress has been made in understanding the role of these membrane transporters in drug safety and efficacy. In particular, more than 400 different members of these transporters have been characterized, and they are majorly grouped in two superfamilies, like ATP binding cassette, ABC, and solute carrier transporters. So if you see like in clinical pharmacokinetics, drug-drug interaction studies have suggested that transporters often work together with drug metabolizing enzymes in drug disposition process. As we know, the historical phasing concept of, yes? Okay. As we know, the historical phasing concept of drug metabolism that was introduced, and it consists of two phases of drug metabolism, phase 1 and phase 2. Phase 1 and the phase 2. With this concept, the biological membrane barrier, that is transporters, which obstruct the accessibility of these a small molecule to the metabolism side, they are not considered. So this two-phase concept then further extended to the four-phase concept. To the four-phase concept where the drug transporters were detected and that guide the drug and drug metabolites in and out of the cells, in particular some water-soluble or the charged drug that are virtually not able to overcome the phospholipid membrane barrier. So for cellular drug process, the term phase 0 and phase 3 was introduced based on the original phasing concept of the drug elimination. So the phase 0 refers, that allows the entry of drug molecule into the metabolism site, while phase 3 allows the exit of the metabolite from the cells. So this concept included under the term ADMET, that's absorption, distribution, metabolism, excretion, and transports. And this issue of xenobiotic elimination is a matter of combined process of both drug transporters and drug metabolizing enzymes. So this phase concept can be explained with a situation in the liver as depicted in this the particular figure. Like here, the sequential steps of drug elimination by metabolism and the membrane transporters. So, and phase 0, here you can say the phase 0, the drug's uptake is taken by different transporters into the hepatocytes, and phase 1 and the phase 2 then comprises of the phase 1 and phase 2 metabolism, where the drug is being metabolized by phase 1 and phase 2 drug metabolizing enzyme. And we can take an example for this one, for the statins. All the statins drugs, I mean, they are taken up by some OATPs into the hepatocyte. They are metabolized by cytochrome P450s as a phase 1 metabolism, and in phase 2 they are getting conjugated by different gluconides and maybe different glutathiones. And these phase 2 conjugates, they are then eliminated with the help of some of the transporters, for example, MRPs and the BCRPs. So this is the this now here complete the both like the transporter and drug metabolizing enzyme role in drug disposition from the liver. So now, I mean, coming to the classification of these drug transporters. So this can be classified based on the mechanism of their transport. So they are generally classified into 3 different categories: uniporter, symporter, and antiporters. So as you can see here, the uniporters are involved in facilitated drug transport, and it works by binding one molecule of the substrate at a time to move it along its concentration gradient. For this, the example is the proton pump in the stomach, and it is passive transport, does not involve any ATP. While the symporter and antiporter, these two are involved in the active transport. So the symporters, as you can see here in this the picture, symporters, they transport 2 molecules in the same direction. And for this, you can say the example is the glucose transport, SGLT1, that transports sodium ion and the glucose together, I mean, while in the absorption of the glucose. The second is antiporters. Antiporters, these are the transport molecule. They, I mean, they work like 2 molecules in opposite direction. They transport 2 molecules in opposite direction. Examples is sodium potassium ATPase, where this it's like it's taking sodium and the potassium as an exchange, So it uses 2 different ions or 2 different molecules doing its function. But these active transports are further, they're being classified as primary, secondary, and tertiary active transport. And they have been divided into 2 big families of the transporters. So the primary active transporters requires ATP dependent hydrolysis as the first step in catalysis. Examples are like ATP binding cassets. So that's termed as ABC, ATP binding cassets. They have like different classification based on their function or their localization. So that includes the P-glycoproteins that's very commonly, I mean, very much explored in the as a transporter and very well known. And then the others are the BSEP and the different isoforms of MRPs, which are the multi-drug resistance associated proteins, and then the BCRP, breast cancer resistant protein. The second class is the secondary or the tertiary active transporters. These are driven by the exchange of co-transporter of intracellular and extracellular ion with the substrate. So these SLC drug transporters do not rely directly on ATP hydrolysis and are largely, but not exclusively, they are the uptake transporters, meaning they take the drug molecule inside the cell, and then further metabolism and the reaction takes place on those particular metabolites or the drug molecules. For this one, these SLC molecules, they are then further classified, and they include a large variety of different transporters, including NTCP, that's sodium taurocholate co-transporters, PEPT, and these are the oligopeptide transporters 1 and 2, and these are the OCTs, organic cation transporters, and OCTN, is novel organic cation transporters and organic anion transporters. And they also include some of the OATP. I mean, this includes the OATPs, which are the organic anion transporters polypeptide. So these overall, all the transporters, they have been classified majorly into these two big classes, ABCs and SLCs. And you can see in the literature that they are like, they use, sometimes there's a common name, which are like NTCPs, organic cation, or sometimes they use, there's a classification name like SLC 22A1, and this you can see in these things in the literature. Coming to the localization, so these drug transporters are found almost in every tissue in the human body. But in drug development, particular attention have been paid to the transporters expressed in the epithelia of the intestine. That you can see here, that's the absorption site of the drug molecule. And the second, in the hepatocytes, that's a metabolism site. And then the elimination or the excretion site in the kidney, that's a kidney proximal tubule. And also in the blood-brain barrier when the drug has been distributed. So this ADME process, like in all these, the complete disposition process, you can see there are the different these drug transporter expressed on the apical and the basolateral, the membrane side from, you can say, the intestine and in the hepatocytes. And there are the numerous, they perform like the function of drug disposition along with these the drug metabolizing enzymes. So coming to the clinical significance, like why we should study these drug transporters. So I will give an example of the P-glycoprotein transporter, which is like very much explored. And so in around 2012 and 13, Dr. Schinkel, he was working on one of the knockout model, mouse model of this PGP. So he received a message from his animal care facility that all of his knockout mice, they're suddenly dying. When the animal caretakers were asked, like, okay, what they have done, what they have done, because why these animals are dying? So their answer was very predictable. Oh, we did nothing. We did nothing. We just followed the regular protocol. So, but during investigation, it emerged that, however, that these mice have been sprayed with a pesticide, that's ivermectin, and to counteract the mite infestation. This ivermectin is a strong neurotoxin, and normally its entry into the brain is blocked by the PGP transporters. So it's an efflux transporter, so its entry is blocked, and entry is blocked into the central nervous system. But a toxicity analysis, later on, the toxicity analysis reveals that in the PGP knockout mouse, this ivermectin concentration is increased 100-fold as compared to wild type, and this accumulation led to the lethal neurotoxicity. So this indicates that the PGP has an important role in the limiting entry of the various drug molecules into the central nervous system. If you take another example of cerebrastatin and gemfibrozil combination therapy, so this combination was used for the lowering the blood cholesterol and triglycerides. But it has been observed that with this combination, the plasma concentration of the cerebrastatin is increased significantly, as you can see in this the PK diagram, like with and without gemfibrozil, the cerebrastatin concentration is significantly increased. And this is due to the fact that the gemfibrozil and its a gluconide metabolite both inhibit OATP1B1 mediated hepatic uptake and CYP2C8 mediated metabolism cerebrastatin. So it's inhibited its drug metabolism directly. So that's why its concentration increased, and this increased exposure of this cerebrastatin in presence of gemfibrozil explains the high incidence of myopathy, and that leads to these severe consequences. And at that time, that gave around 52 deaths in that the clinical trials. So coming to one more examples of drug-drug interactions between these transporters, OATP1B1 transporter and So again, with the statins, like inhibition of this OATP1B1 mediated hepatic uptake, this appears to contribute to the significant increase in the static concentration in blood after the cyclosporine, because cyclosporine is an inhibitor of OATP1B1. So as you can see here in this particular figure at the left hand side, the AUC, the area under the curve, you can say the plasma exposure is increased from 5 to 20 fold, and that's led to the serious consequences. So in addition of the drug interactions, OATP1B1 transporter polymorphism is also associated with increased drug exposure of these particular statin molecules. As you can see here, from 1.5 to 3-fold increase in drug exposure, and that led to the side effects. Apart from these statins, OATP1B1 also have been shown to increase the exposure of other drugs such as rapaglinide, irinotecan, which is an anti-cancer, and ezetimibe drug molecule. So now, like, I'll give a brief about the inter-individual variability, how these drug transporters is variable from one individual to another individual, and these affect the final the clinical significance or the final outcome of those particular drug molecules. So this variability can be at two different levels. One, like there is a polymorphism, there is a single nucleotide polymorphism that because of that, the activity of those particular transporter differ from individual to individual. And the second case might be like if you have the quantitative abundance or the expression level of these transporters might be different from one individual to another individual. And that might lead to the differences in the drug disposition. So here, these examples is from the, again, from the OATP1B1, but this is because of the polymorphism in its, because of polymorphism at particular site. So OATP1B1, as we have seen in the previous example, is an uptake transporters expressed and in the human hepatocyte, and OATP1B1 has a broad specificity, is important in both systemic and the liver exposure of many drug molecules. So the genetic variants with decreased activity may increase the systemic exposure. So as you can see here, the genetic variant with the CC type that have decreased activity with respect to 1B1 variant, 1B1 transporter. So you can see it has a decreased activity. So exposure on here also on the right hand side figure, exposure level for particular drug, for example here the statins, you can see the exposure level is significantly increased, while the genetic variant with increased activity, that's 521 TT genotype, this one having very low plasma exposure. So you can see the side effects of the statins as a myopathy, and this was like published in the New England Journal of Medicines, which is very reputed journal. And you can see here like the cumulative percentage of the patients who have had myopathy is high only with the CC genotype, while with the other one, the TT genotype, which is having the high activity, you don't have, because, and plus here in TT genotype, the concentration are very less. I mean, the plasma concentration are very less, and that can be to the subtherapeutic levels. There might not be having any physiological benefit in these particular individuals. So like you can see now like how the difference in the expression of these particular transporters affect the final outcome of the drug. So here another example, like we can study this variability in the expression of this kidney transporters by the you can say the quantitative proteomics. So here what we did is like from different human kidney samples, we processed and we quantified different drug transporters. You can see on the x-axis, and these are like compared. So you can see here there is almost 10-fold difference in the total expression level. So this is not a genetic variant. This is like all are other than the genetic, there's expression level might be different, and this expression level, this variable expression of these drug transporters may lead to variable drug disposition, leading to either drug toxicity or the loss of efficacy in drug development. So if we, coming to the variability from preclinical model to humans. So if we talk about the cross-species variability in drug transporters and its significance. So during drug development, the FDA requires like in vivo repeated dose toxicity studies of a drug candidate to be conducted in at least 2 animal species, and one of them must be a non-rodent. So before initiating the human studies, so typically these species are the mouse or rat and dog or monkey. So to interpret the toxicity generated in animal and predict their clinical relevance, it is really important to understand the potential interspecies difference in the mechanism of the drug toxicity. Like first we have to know like how these drug transporters are different from preclinical species versus human, like which particular model, like either human or either mouse or rat or the dog is more close to the human in terms of the drug transporter functions. So this is an example of the phyalurидine. This is a phyaluridine toxicity in a human. This is a nucleoside analog, and it was investigated as a potential therapy for hepatitis B virus infection. So in a clinical study, there is unexpected toxicity happened, and that led to 5 out of 15 death of 5 out of 15 patients from the liver failure, and then further 2 more patients required the liver transplantations. And this toxicity was very unusual because it was not detected in any of the animal studies. So, before going to the human trials, the phyaluridine was tested in multiple animal models, and it did not show any toxicity in any of the animal model. The reason phyaluridine is toxic in human is because we have a unique drug transporters in the mitochondria that's called equilibrative nucleoside transporter, ENTs, that mediate the transport of the nucleoside, nucleobases, and the therapeutic drug molecule, which are having the similar structure. So it allows the entry of these drug molecule inside the mitochondria. And as we know, the mitochondria is the powerhouse of the powerhouse, and you can say that generates energy. So once this phyaluridine enters into the mitochondria, it shut down the powerhouse and energy generation, and that leads to the cell death. But these particular transporters, when they were doing the study in the preclinical species like mouse, rat, and even dog, so they did not find these particular transporter in the mitochondria. So that's why this toxicity was not seen in the preclinical species, but it's seen with the humans. So there is another example of the variability across preclinical species versus human. So the tenofovir is antiviral drug as used it's an acyclic nucleotide analog of adenosine monophosphate that inhibits HIV reverse transcriptase. So tenofovir long-term use is associated with the kidney toxicity. So this is exactly the same example for in preclinical species from the mouse, rat, and monkey, even monkeys, which is considered as very close to the human. This repeated dose toxicity of tenofovir was not mentioned or not described before. So as you can see here in this particular picture, the tenofovir shows the toxicity in kidney. So it from the blood, it is taken up by OAT1 drug transporters, and in proximal tubule, and it is from proximal tubules is taken out by MRP4 drug transporter. So generally in the kidney, 2 different transporter uptake and the efflux transporters, they work in tandem to get this particular drug molecule out of the body. So like generally, like these are the on the basolateral membrane, there is a lot of uptake transporters. On and apical membrane, there is a lot of these are the efflux transporters. So the drug molecule from the blood is taken up by the uptake transporters, come to the proximal tubule, and then it's taken out by the in the urine by the efflux transporters. So now you consider a situation like, for example, a drug may be rapidly cleared from the kidney in one animal species because of high expression of this drug transporter. As a result, kidney of the concentration of this particular drug will be low. In contrast, in another species, for example, different preclinical species, which has much lower expression of the same transporters, may demonstrate elevated kidney concentration, potentially causing nephrotoxicity. Therefore, I mean, it is really important to compare the expression of these drug transporter in tissues of the human and the animals routinely used in toxicological studies. So what we did, we quantified these O81 and MRP4 concentrations in the kidney cortical tissues from 5 different species: human, monkey, dog, rat, and mouse. And you can see here, these concentration, they are quite variable between preclinical species versus human. So always, like when we are looking at drug disposition, so along with the drug metabolizing enzyme, drug transporters, they also play a very important role in terms of drug disposition, leading to either toxicity or the subtherapeutic concentration of these. So, I mean, coming to some of the application, like how application of these drug transporters, how these drug transporters can be utilized as to increase our drug acceptance or for the drug development. So the first application we can see here is to improve the bioavailability. So when we can exploit the function of this transporter to improve the drug bioavailability. So this is an example like ganciclovir. This is administered orally and intravenously to treat the prevent CMV infection, that is cytomegalovirus infection. And this particular molecule, ganciclovir, has very low or very poor oral bioavailability, which is around 5 to 8%. So they have another molecule which is synthesized as a valine ester of this particular, that's called valganciclovir. And then this molecule shows almost tenfold high bioavailability, because this valganciclovir is a substrate for the PEP1 transporters. So because of this transporter mediated uptake, this oral valganciclovir exhibited almost tenfold higher plasma concentration compared to the oral formulation of ganciclovir. So we can exploit like this function of these transporters to increase the bioavailability of some of the drug molecule. So coming to the second application, the second application of this particular, we can use these transporters as a drug target. So as we know, kidney plays a crucial role in glucose homeostasis. And you can say almost around 180 gram of the glucose is filtered every day. During this glucose homeostasis, the reabsorption of glucose in the kidney is mediated by one of the transporter called SGLT2, which is a sodium glucose cotransporter. And this is expressed is on the PCT, proximal convoluted tubule, and you can see here this absorbs almost like around 90% of this. The glucose is absorbed at this particular region, in the kidney. But so the new approaches are coming if we can block this particular transporters activity. So there are the inhibitors, and people are working on the inhibitors that can block the function of the SGLT2 transporter, leading to the loss of this the glucose into the urine. Loss of this glucose into the urine that can be used as to reduce the blood glucose concentration, and they have been successful doing. These particular compounds have been successful and coming as a new therapeutic molecule to treat type 2 diabetes mellitus. Importantly, this does not affect the insulin action and secretion, in which are the common targets of existing type 2 diabetes medications. Apart from reducing this glycemia, these classes are reported to have the beneficial effects of cardiovascular and the renal system, such as weight loss and the blood pressure reduction, which are commonly associated with diabetic complications. Coming to the last application of the transporters, we can exploit these transporters in precision medicine approach, like to increase the drug efficacy. The example is gemcitabine efficacy. So this gemcitabine monotherapy is standard chemotherapy for the pancreatic adenocarcinoma. And this gemcitabine requires plasma membrane nucleoside transporter protein, that's the same ENT. These particular transporters to efficiently enter the cell and exert its toxicity, because and this particular transporter have been found on the in the cancer cells. Once this allows the entry of this particular drug molecule, this drug molecule can exert its toxicity in that particular cancer cell. So in vitro studies have demonstrated that if there is deficiency of this particular transporter, it is most widely abundant and distributed in the human cell. If there is a deficiency, the resistance of this to this gemcitabine therapy happens, like because of the deficiency, you see the resistance to this particular drug molecule. So the patients with the pancreatic adenocarcinoma, if you see, like if they have this transporter uniformly distributed or detected by the immunostaining, they have significantly longer survival as compared to the patient where there is a deficiency of this particular transporter on the cancer cells. So we can use these as a precision medicine approach, meaning like through the immunostaining techniques, we can identify those patients they have the higher abundance of these higher expressions of these transporters or the lower expression, so we can modify the dose of this particular chemotherapeutic agent. So we cannot have like the overdosing or the underdosing. So we can design like individual dosing for these the cancer patients. So with this, like I'll just come to the last slide. So take home message, like what we learned from this particular presentation is like clinical pharmacokinetic drug interaction studies, they have suggested that the drug transporters, they often work together with drug metabolizing enzyme. So only in eliminating any drug molecule, only drug metabolizing enzyme does not play. There is a role of these drug transporters, and they both work, drug transporters and drug metabolizing enzymes. They both work in tandem, they both work in concert to get rid of this drug molecule from the body. So the consideration must be given to the role of drug transporters in ADME process of new chemical entities in the drug development. And we have seen, like with a couple of examples, there is high inter-individual variability, meaning variability from one human to another human, and also interspecies variability from preclinical model to the human. So these variability are really challenging in drug discovery. So we need to understand and we need to take into account this variability while developing a new drug molecule. And at the last, like we can exploit these transporters as a target for the drug development, we have seen some examples, like we can improve the oral viability of the drug molecule, or we can use them as a target molecule for to gain some therapeutic some therapeutic interventions. So with this, I'll stop and I'm ready to take any question. Thank you for your patience and listening.
Thank you, Dr. Abdul Basit, for your session on drug transporter, case for inter-individual variability in drug exposure. Now let's take a few questions posted. I think I cannot see the question in the chat box. So it's okay. I think the participant, if you want to ask the question, you can unmute yourself and you can ask the question.
Yeah. Okay, if you, I mean, if you have any questions, you can ask, or you can email me later on anytime if you need any, I mean, particular questions you want to ask.
Dr. Abdul Basit, this is Dr. Sharik
Yes.
here. In my experience in drug development, essentially, PGP comes on the first hit, where we talk about more into efflux transporters and that affecting variability. Have you seen any other evidence of, let's say, OATP or other transporters that have appeared in the label of drugs where they talk about the variability that has been accounted because of these transporters, or the genetic variability in different populations of these transporters? In your experience, have you seen anything of that? Can you?
Yeah, yes, I mean, as I've given one examples of OATP1B1 is one of the transporters, like is highly variable because of its you can say genetic variability. They have different variants, different single nucleotide polymorphism. So OATP1B1 is very, I mean, well known in these particular things. Like, and very, you can say if people have this variability, the statin therapy, you have the lot of side effects for the statin therapies. And other than the polymorphism, the variability we are looking at, I mean, all these drug transporters in the human with respect to like the age, or the you can say the ontogeny, how the I mean, these levels of or the expression levels of these drug transporters are different from, you can say, from the neonatal to the adults. So we have seen there are like around 10-fold difference in a couple of those transporters. So I mean, as I've seen, I've given that one particular the slides, but those are only in adults. But in the literature, there are a couple of examples. For example, OATP1B1 is because of polymorphism, and some MRPs we recently we published in 2019, yeah, last year, about some of the MRPs, like they have some variability with respect to ontogeny, while I mean it's getting while you can say we from infants to the adults. So that comes a picture like maybe like the same drug molecule which is being transported by this some of the MRPs, which are the efflux transporters. So you have to consider those things like the age of the patients, like the expression might be different, and the dose of that particular drug molecule might be like adjusted based on those age.
Thank you. Thank you. I think that was helpful. Yes. Yes, Professor Abu Sufyan, we can take any other questions from the audience.
I think there is no question, sir. So,
Okay.
shall we proceed
Yeah,
ahead?
yeah.
Yes, yes.
Okay, okay. So, thank you, Dr. Abdul Basit. Now I request Dr. Sharik, Dean, School of Pharmacy, to summarize today's session and give concluding remarks.
Thank you. Thank you. I think in the interest of time, I think it's already fair. We've shooted a little bit over time here, but I really would like to thank both of our speakers, Dr. Farooq Sheikh and Dr. Abdul Basit, for giving a very nice overview about two important areas. One of them is innovative disease models, and the other is more into the whole area of transporters. That is, a huge area of lot of interest with regards to, drug transporters, with regards to, PD transporters. So a lot of variability, and in my experience during the clinical stint at BMS, we used to interact very regularly with the preclinical folks, and there's a huge amount of interest that's there in terms of developing the different genetic variants of these transporters and actually assessing what is the role of that. So thank you both of you essentially for a very informative and a very a nice session. Dr. Basit, I know it's very early morning for you. I'm not sure if it's snowing out there. I can see white background for you. And both for Dr. Farooq also, so for giving their valuable time. Thank you so much. I think it's a fairly interesting session, and we look forward for more such interactive sessions with you, both of you. Thank you once again. Over to you, Professor Abu Sufyan.
Thank you, Dr. Sharik, for summarizing and giving concluding remark for a day. We are done with today's session. Feedback and attendance link form we have already released in chat box. Please see that you give that feedback in 20 minutes. It will be active, remains active for 20 minutes. Our tomorrow session will start sharp at 5 p.m. Please note that Zoom attendance link for a day 2 shall be shared in workshop WhatsApp group. Those who have not yet joined this group are requested to please join this group for further smooth communication. See you tomorrow. Have a nice day. Stay safe and stay blessed.
Thank you. Thank you. So just before we close it up, we have a very exciting session coming up tomorrow where Dr. Nafis will be talking about more into optimizing of drug targets, something that Dr. Farooq alluded today with regards to, more of a rational-based drug discovery. So he'll be talking about more about molecular docking and his experience at Monash Lab. Thank you. Thank you once again. A very a good evening and a good night to all of you, and good morning to Dr. Abdul Basit. Thank you so much.
Thank you.
Welcome.